All right, hi everyone. This is lecture 11, the second lecture of our cardiovascular portion of this course, and it's going to cover blood vessel anatomy and blood. When you're finished with the lecture, please check the learning objectives to make sure that you've gotten everything out of this lecture that we've intended. A brief review of the cardiovascular system. We covered the anatomy of the heart in lecture number 10. The heart is a double-sided pump that establishes blood pressure. The blood vessels then carry that blood, and the blood is a liquid connective tissue that transports dissolved and suspended materials. Blood vessels are a network of layered tubes acting in a closed circulatory system with the heart. They carry blood to and from the heart. Blood vessels are largest near the heart and branch out towards the target tissues. If you're starting at the heart, the arteries move away from the heart. So here we've just drawn the heart as a box. But this would be the left ventricle, which is pumping out of the aorta. And starting with the aorta as the largest of the arteries, the blood will move away from the heart. And those arteries will then branch into elastic and muscular arteries, and then arterioles, which are small arteries. As they get closer to their target cells, they will become microscopic capillary beds, where diffusion at the tissues will take place. And we've already calculated in previous lectures the net filtration at the capillary beds, where the exchange of fluids occurs. After that exchange, the blood will return to the venules, which are small veins, and move progressively towards the heart as the vessels get larger and larger. And we have large veins then returning blood towards the heart. So we remember this as saying arteries away. So arteries with the A, arteries move away from the heart, and veins always move towards the heart. We will also briefly cover the role of the lymphatics. We'll begin by looking at the histology of blood vessel walls. If we take a very general approach to look at what generally is present in each of the blood vessel walls, we start with the lumen, or the space in the middle where the blood travels. And then the layer closest to the lumen is the tunica intima, which is the endothelial layer. There's a small amount of basement membrane, and in arteries there's an internal elastic membrane. That's in muscular arteries only. After that, we have the layer of smooth muscle, which is the tunica media. There will be an external elastic membrane for muscular arteries here as well. Finally, the last layer of the blood vessel wall is the tunica externa, which is also referred to as the tunica adventitia. This is fibrous connective tissue with collagen, and it has its own layer of internal blood vessels, the vasa vasorum, and its own set of lymphatics. I love this image from your textbook. And what we're going to do now is go through each of the types of blood vessels and review the specifics for each type. Before we do that, I'd like you to pause here and look at this diagram carefully and highlight the differences in blood vessel structure. Okay, can you see some differences? I've highlighted them for you here as well. So if you look at the arteries, you'll see that the large arteries contain elastic tissue. And then if you look at the veins, you'll see that the veins contain valves. Because veins are low pressure, they need those valves. We'll talk about those in a moment. I also want you to notice the thickness of the layers. It's much thicker in the arteries, particularly the tunica media, compared to the veins, which have very thin walls. And look at the capillaries. The capillaries really only have an endothelial layer and a tiny bit of basement membrane, so they don't have all three layers like the rest of the blood vessels. Okay, let's look more closely. Here's some blood vessel histology for you to return to after we go through these. So arteries move away from the heart, and they're going towards the tissues to deliver oxygenated blood and nutrients to the cells. They have thick elastic walls and a large radius. They're highest pressure nearest the heart, where blood is being pumped out. The large arteries expand to absorb and store the pressure of the pumped blood leaving the heart. We'll talk about this when we lecture on blood pressure, so keep this in mind. The arteries have this very important stretch capability in order to allow that pressure of the pumped blood to continue from the heart and move towards the tissues. Because they're high pressure, they also need to be strong, so they have a lot of collagen for tensile strength. So remember when we talk about blood pressure that artery pressure fluctuates with the systolic and diastolic events in the heart. The largest of arteries are the elastic arteries and the muscular arteries. The elastic arteries expand and recoil. That's to maintain continuous blood flow and even out blood pressure as the heart ejects blood. The muscular arteries are medium in size. They have the most smooth muscle, and these are typically the arteries that we would name in the lab. Arteries like the brachial artery, like the subclavian artery, the femoral artery. These are the large muscular arteries. They distribute blood, and they have slightly less recoil and slightly less elasticity than the elastic arteries. The arterioles are small arteries. They're mainly smooth muscle, and they have sympathetic nervous system innervation, as do all of the other arteries, but they're especially responsive to this innervation because they're very high resistance. So in response to this nervous system innervation, they can slow down the flow of blood from the arteries to decrease pressure and regulate blood flow to the tissues. That is, when the sympathetic nervous system activates the smooth muscle of the arteries, it's going to constrict the smooth muscle by contracting it, and that closes the diameter of the blood vessels, reducing the size of the lumen in order to slow down blood flow. That happens locally in the tissues. When they're relaxed, when the sympathetic nervous system innervation is less, these vessels will open, and that is vasodilation, and that's done to increase local blood flow. This is the vasomotor response. Vasoconstriction is contraction of smooth muscle, particularly in the arterioles, to decrease the diameter of a vessel. This increases the resistance and can slow or stop the local blood flow. Vasodilation is the opposite, and that's less sympathetic nervous system activity, which relaxes the smooth muscle in the arterioles to increase the diameter of the vessel. This opens the diameter of the vessel for less resistance and increases local blood flow. I'm emphasizing local here because when we get to blood pressure, we're going to talk about the difference between local regulation of blood pressure and blood flow and systemic regulation of blood pressure and blood flow, and they're quite different, so keep that in mind. This vasomotor response can be triggered in any smooth muscle, but it has the greatest effect on the arterioles. Now we get to the microscopic capillaries. These are the blood vessels that are the sites of exchange with the tissue cells. They're thin-walled, and they branch out densely at the tissue sites. No cell is less than 0.1 millimeter from a capillary. They are low blood pressure to allow slowing of the blood flow such that the tissues are not damaged with this blood flow. The exchange will occur through diffusion, and that is through the endothelium layer. Permeability will vary by how tight the endothelial cells are joined together, and there's three main types of capillaries. The most common are the continuous capillaries. Here we have an image of the different types of capillaries. So in general, you can think of the continuous capillaries when you're thinking of a capillary bed. And then in particular tissues, we'll talk about these other exceptions of permeability. Fenestrations are windows. So fenestrated capillaries have little window-like openings and are high permeability. We see these in the kidney, the small intestine, certain endocrine glands. These have large pores for filtration and absorption. We also have sinusoid capillaries, which have even larger permeability openings. These are found in the liver, the bone marrow, the spleen, and the adrenal medulla. They have these large clefts and pores, and larger molecules and cells can pass through these sinusoids. When we get to the kidney and when we get to the liver, we'll talk about the function of the capillaries specifically in those tissues. It's also important to think about microcirculation at the capillary bed. So between the arterioles and the capillaries are meta-arterioles, and these have little sphincters. A sphincter is a ring of smooth muscle that can be contracted to cut off flow to an area. So here we have the arteriole, and here we have the capillary bed. And there's a sphincter which can be opened to allow a larger amount of blood flow through the capillary bed. Or it can be tightened to cut off that blood flow and be shunted. This would be done in tissues that have less metabolic need. So you would open up the precapillary sphincters to increase the blood flow to an area, or you can close the precapillary sphincters to shunt the blood when less blood flow is needed. From the capillary beds, the blood will return to the heart through the venules and then the veins. The venules are small veins leaving the capillaries. They have thin walls and a small radius. The veins are the larger vessels returning blood from the capillaries back towards the heart. They're much thinner walled than the arteries. They also have sympathetic nervous system innervation, but because of their large size and because of their lack ability to recoil and their low pressure, this innervation has less of an effect than it does in the arteries and arterioles. They're easily stretched to retain blood, but they're not elastic, so they do not recoil. They have slow blood flow and low pressure, so they require the movement of skeletal muscles to help pump blood to the heart. They also contain valves. Here you can see the valve in a vein where the blood would go towards the heart, and if the pressure got too low, the valve would prevent backflow of that blood and keep the blood flow going towards the heart. There are some large flattened drainage veins called venous sinuses. These are large drainage areas. We'll learn about the dural sinuses in the brain, and we've already touched on the coronary sinus in the heart. Again, veins have valves. These are part of the tunica intima and similar to the semilunar valves of the heart. Varicose veins are abnormally dilated veins due to valve failure. In a chronic condition, there's chronic venous insufficiency. Impaired venous return can lead to venous congestion and edema, which is caused by, ultimately, this chronic damage to the valves. This will lead to poor circulation and edema. You get thin skin in these regions, discoloration, and poor healing due to lack of waste removal and insufficient blood flow. Before we end the portion on blood vessels, I want to talk about the endothelium. The endothelium is becoming more and more appreciated as a tissue almost on its own. It's a very active tissue. And so some are starting to classify the endothelium separately from the other layers of the artery wall. So it's found within the tunica intima, and more research is looking at the synthesis and release of molecules specifically from the endothelium. These molecules can help to regulate coagulation and related processes. They're looking at these in terms of immune system function, tissue growth and wound healing, and vasoconstriction and vasodilation regulation. So again, the endothelium is the innermost layer of all of the blood vessels, and it's releasing these important molecules and responding to changes in the local environment. Here's an example of endothelium regulation of vasodilation from your textbook. Ultimately, this leads to the release of nitric oxide. So the endothelium directly regulates and releases substances for vasodilation. Some example triggers are shear stress, platelet-derived factors, exogenous substances, and prostacyclin. This will lead to the activation of an enzyme, nitric oxide synthase, and the release of nitric oxide. The effect of nitric oxide is to relax the smooth muscle in the walls of the vessels, and that vasodilation opens up the lumen of the blood vessels and increases local blood flow. There's also regulation of vasoconstriction. So the endothelium directly regulates and releases substances for vasoconstriction. One of the key molecules here is angiotensin II, although there are others. So some example constrictors are arachidonic acid, certain metabolites, and endothelin. Finally, there can be metabolism or breakdown of bradykinin, which is a vasodilator. Any of these different molecular pathways can lead to vasoconstriction. The endothelium has also been found to be important in the regulation of platelet aggregation. The endothelium releases substances that affect clotting and inflammation, and it can go in either direction, increase or decrease, depending on the signals present. It can affect the number of platelets adhering to the vessel wall, and it can affect the recruitment of inflammatory cells. Here's some examples of factors that are related. We're going to cover coagulation and platelet aggregation in a future lecture. This box from your textbook talks about the balance of vasodilation and vasoconstriction factors. In particular, it highlights the effect of high angiotensin II, for example, in high blood pressure or shear stress, and low nitric oxide. In this case, the balance is disrupted, and you're going to be pushed towards vasoconstriction. Treating with ACE inhibitors can reestablish that balance and regulate the vasoconstriction to keep the blood pressure from getting too far tipped towards high blood pressure. Blood vessel growth also occurs as an effect on the endothelium. So new vessel growth occurs in response to wound healing and injury. For example, the endothelium can release growth factors, such as this one, vascular endothelial derived growth factor, which leads to angiogenesis. There's actually 3 types of blood vessel growth. Angiogenesis, which we have already talked about in previous lectures, is growth of the capillaries. Arteriogenesis is growth of the larger arteries. And vasculogenesis is just general vessel growth from stem cells. This is of interest to oncology research, where they're looking at the factors secreted by the endothelium, such as vascular endothelial-derived growth factor, or VEGF, and the similarities of this signaling in tumor cells. And the hope is that a better understanding of the angiogenesis in the normal endothelial cells and angiogenesis in tumor cells may help with future research on how to block tumor growth. This is a diagram which shows the amount of blood held in any of the vessels at any one particular snapshot in time. And the key takeaway here is that because of the low pressure, the veins are a reservoir for blood. So most of the blood at any one time in our bodies is being held in the veins. And this will be important when we calculate blood pressure and mean arterial pressure later. So we've talked about the arteries, the capillaries, and the veins. The last set of vessels we need to consider are the lymphatics. So lymphatic vessels are similar in structure to veins, and they pick up excess fluid and large molecules from the capillary beds and return this excess fluid and large molecules to the venous system. The biggest difference between lymphatic vessels and veins is that it's a one-way system. So if you look at a microscopic view, you'll see that we call these blind-ended vessels, meaning that they're closed at one end, and they have these sort of flap-like valves. So they're closed at one end, and they have these flap-like valves that will open and allow the large molecules and the fluids to enter, but they only go in one direction because that blind end keeps them from going back into the tissues. So the fluid and the molecules will enter the lymphatic vessels and be pushed back up towards the heart. There's no pump in the lymphatic vessels, so they rely on skeletal muscle action to be pushing the lymph back towards the heart. They're very low pressure, just like veins. So they also have valves, like veins do. They're permeable to large proteins and particles. And so this can clean and remove these substances from the interstitial space. This fluid, the lymph, will travel through the lymphoid tissues, including the lymph nodes and the spleen, and we'll cover the lymphoid tissues when we cover the immune system later in the quarter. Just to keep in mind that the lymph is made up of primarily dissolved proteins in water, lymphocytes, and other immune cells. Okay, we're going to pause here to review the blood vessels and the lymphatic vessels before we move on to the blood. So take a moment to make sure that you understand the differences between the different branches of the arteries, the capillaries, the different branches of the veins, and the lymphatics. Okay, let's move on to blood. Blood is distributed to and from the tissues through the blood vessels in closed circulation, driven by the pressure established by the heart. There are many functions of blood. Blood distributes oxygen and nutrients. It removes waste. It brings hormones and other important molecules to target organs in the body. It also regulates. It regulates body temperature by absorbing and distributing heat. And it carries ions and acids and bases for electrolyte and pH regulation. Blood also regulates fluid volume for circulation. Finally, blood contains many white blood cells, which are the key cells in the immune response. There's also blood clotting, which prevents blood loss, and injury response and healing. So blood is a liquid connective tissue with cells suspended in the plasma. We're going to go through the components of blood. We'll start with plasma, and then we'll go through the cellular elements: red blood cells, white blood cells, and platelets. When you're done with the lecture, come back and look at this blood smear. This has some examples of all of the white blood cells in addition to the red blood cells and some platelets. Let's start with plasma. So plasma is the liquid portion of blood. It makes up about 52 to 55% of the total blood volume. I'm going to give you percentages for the plasma and all of the formed elements within the blood. Keep these in mind. You're going to do lab tests later that are going to help you to determine when these different percentages are out of balance. So blood plasma is about 90% water, about 6 to 8% protein. We've already talked about the importance of albumin protein. There are also globulins, fibrinogen, and a small percentage of other proteins. It's about 1% electrolytes. There's also nutrients like glucose, amino acids. There's vitamins and lipids. There's waste, dissolved gases, and hormones. Blood functions as a buffer. Because plasma is 90% water, this actually itself acts as a pH buffer, resisting changes in pH. Osmotic balance is important, as the composition of water and electrolytes can be balanced to maintain osmotic gradients. There's also the importance of the protein and the oncotic pressure, as we've discussed when we covered net filtration. Blood proteins can carry hydrophobic substances, and that can transport certain molecules. Others can be dissolved in the plasma. Plasma proteins have many functions and are important in osmotic balance, immune defense, transport of molecules, and blood clotting. One way to look at the protein content of blood is to do serum protein electrophoresis. Here's an example of the raw data that you would get from running these proteins on electrophoresis gels. And here's an example of the plots you would get from the amount in each of those bands. So a darker band here leads to a bigger peak in this graph, and the lighter bands will lead to smaller peaks that are measurable as you analyze that electrophoresis data. So starting with the albumin as being the most common and highest peak when you do this kind of analysis, it's about 57% of the total protein. 4.5 grams per deciliter is common. It's the most abundant protein, and it's produced by the liver. It helps to maintain colloid osmotic, also called oncotic pressure. It's a carrier molecule as well for lipid-soluble molecules, certain drugs, and hormones. Next would be the globulin proteins. They're about 38% of the total protein, about 2.5 grams per deciliter. They're produced by plasma cells in the lymphoid tissues, and they're classified by this plasma electrophoresis. There are different classes of globulin proteins. There's alpha 1 and 2, beta 1 and 2, and gamma. They're important for immune defense. Looking at globulin proteins is one way to analyze certain conditions. We know that the balance of globulin proteins changes, for example, in multiple myeloma, and we'll talk about this later. Fibrinogen is also common. It's about 4% of the total protein. It's a clotting factor and a precursor for the fibrin portion of clots. All right, now let's look at the cellular components of blood, starting with the red blood cells. About 4.2 to 6.2 million per millimeter cubed. That's 42 to 48% of the total blood volume. That's a huge number. And the percentage of red blood cells to whole blood is the hematocrit. The amount of blood cells, red blood cells, determines the thickness or the viscosity of the blood. So if your hematocrit is too high, then you can have a greater thickness of blood. There are other determinations of blood viscosity, but the hematocrit is a major contributor. When you look at red blood cells, you'll see that they have a biconcave or a donut type shape. So this 3D reconstructed image here shows the red blood cells quite nicely. What you can see is that the outer wall or the outer portion of the red blood cell is a little bit thicker than the center of the red blood cell. That's why we say they look like a slightly filled donut, where the edges are wider and the center is thinner. So if you look at a red blood cell from the side, it would look like this. So in other words, there's a concavity to the center of the red blood cell. They're also slightly flattened, and they're very small, 6 to 8 microns in diameter. They need to be small to be able to squeeze through the capillaries, and in fact, they can deform even smaller to fit through even the tiniest of capillaries, up to 2 microns small. And as they squeeze through the capillaries, they can then regain their shape afterwards, and we call this being reversibly able to deform. There's no nucleus in the red blood cells, and there's very few organelles. They have no mitochondria, for example. Because they have no nucleus and few organelles, that makes the lifespan of red blood cells very short, about 80 to 120 days. They also contain structural proteins, but the major component of the red blood cell is hemoglobin. So they're really just like bags of hemoglobin. So what is hemoglobin, and why is it so important that we've packed our red blood cells full of it? There's 4 subunits to hemoglobin, and the purpose of hemoglobin is to carry oxygen. So hemoglobin is an oxygen-carrying molecule. It contains 4 pigment groups, each with an iron. This pigment is called heme, and it contains 4 polypeptide chains. That polypeptide is called globin. So there are 4 heme groups and 4 globin chains. We put that together, and we have this 3-dimensional structure of hemoglobin. Turns out there's 2 different types of globins. There's alpha and beta globin chains. Ultimately, what this means is that you have the ability to carry 4 oxygen molecules for every 1 hemoglobin. So hemoglobin is carrying oxygen. 98.5% of blood oxygen is carried by hemoglobin, and that's because oxygen dissolves poorly in the blood plasma, dissolves poorly in water in general. So only about 1.5% of our body's oxygen is carried in the plasma. The rest needs to be bound to a carrier, and hemoglobin carries oxygen quite well. The oxygen molecules will bind to the iron within the heme group, giving each hemoglobin the carrying capacity of 4 oxygen molecules. This binding is reversible so that oxygen can be released when it reaches target cells. When hemoglobin is bound to oxygen, it forms oxyhemoglobin, which is bright red in color. When it has less oxygen, or when it is deoxygenated, it is dark red or bluish in color, and that's deoxyhemoglobin. Hemoglobin can also bind carbon dioxide. It's at a different location than the oxygen binding, but about 10 to 20% of the blood carbon dioxide is bound to hemoglobin. This will become important later as we discuss the hemoglobin oxygen saturation curves. And we're going to do this during our discussion of respiratory physiology. But when carbon dioxide binds to hemoglobin, it forms carb-aminohemoglobin. So one note here that this is a small percentage of carbon dioxide binding. The majority of carbon dioxide is actually carried within the plasma as free bicarbonate. And that's more like 70 to 80% of the dissolved carbon dioxide. So again, red blood cells have a short lifespan. So that means that something needs to be done with the old and damaged red blood cells. So old and damaged red blood cells are degraded by the macrophages of the spleen, the liver, and the bone marrow. The iron from the heme groups is taken up by macrophages in the spleen and liver and lymph and stored for reuse. The globins, the proteins, will be broken down into amino acids and recycled. The heme portion will be degraded to bilirubin. That will be sent to the liver for excretion in the feces through the bile pathways to the small intestine. In the case that something in this degradation pathway has gone wrong, and you either have too much bilirubin building up or not enough excretion of bilirubin, this can cause jaundice. So bilirubin is a breakdown product of heme in the red blood cells. Increased breakdown of red blood cells or decreased elimination by the liver can cause excessive bilirubin levels or hyperbilirubinemia. Bilirubin has a yellow color and it adheres to connective tissue, especially the skin and the white of the eye, which is the sclera. Yellow-toned skin and/or sclera is jaundice. During our discussion of the digestive system, when we cover the liver functions in more depth, we'll return to this concept. Now let's talk about the white blood cells. White blood cells are leukocytes. The normal total white blood count is 5 to 10,000. That's less than 1% of total blood volume. Remember that red blood cells were in the millions. White blood cells, unlike red blood cells, are complete cells with nuclei and organelles. That means when they stain them with typical stains, they stain purple. They're abundant in the lymphoid tissues and they circulate in the lymph and the blood. There are 5 types of white blood cells. Histologically, we can look at the appearance as being grainy or less grainy, and we will then classify them as granulocytes or agranulocytes. So the granulocytes look grainy because they contain enzymes in these granules and digestive vacuoles that help to degrade debris and destroy pathogens. The granulocytes are neutrophils, eosinophils, and basophils. The agranulocytes look less grainy. They have fewer granules visible when they're stained, but they may have enzymes and vacuoles present. The agranulocytes are lymphocytes and monocytes. The white blood cells are very important, so we're going to go through each of the white blood cell types and talk about their function and their appearance. First, neutrophils. We've already heard a lot about neutrophils as we've discussed acute inflammation and other pathological processes. Neutrophils are the most common of all of the white blood cells. So they're 57 to 67% of the total white blood cell count. And that is approximately 3,000 to 5,800 per millimeter cubed. Their appearance is that they are granular, so they're granulocytes. And if you look at a white blood cell under the microscope, in general, we're going to look at the appearance of the nucleus and the cytoplasm. So in a neutrophil, you can see that there are granules within the cytoplasm. And if you look at the nucleus, it's a very unusual nucleus. It's not a single circle. In fact, it's multi-lobed. So here we have 1, 2, 3 lobes in this neutrophil. So it's multi-lobed, and it has a pale red and blue granules within the cytoplasm. Generally, you have about 2 to 5 lobes per neutrophil nucleus. Because of this, we call them polymorphonuclear. Polys, PMNs. Immature neutrophils are called bands. Mature neutrophils are called segs. Lots of different names for neutrophils, all same. Neutrophils are phagocytes. They're the first responders to infection and stress. They're highly present in acute inflammation, where they migrate to the sites of injury, engulf and destroy bacteria using lysosomal enzymes in their granules, and they're going to ingest and destroy debris, and then they'll die within 1 to 2 days. In this process, neutrophils can also release free radicals, and we've talked about the damage that can cause as well. Next are lymphocytes. Lymphocytes come in different forms, and we'll discuss these more carefully when we talk about the immune system. But you'll also hear lymphocytes being referred to as B cells. There are also T cells and plasma cells. They're about 25 to 33% of the total white blood cell count in a normal patient. That's about 1,500 or 3,000. They are very common, so they're easily visible, but they have few granules and no digestive vacuoles. They have a single round nucleus with very little cytoplasm. So already you should be able to tell neutrophils from lymphocytes just based on their nucleus. And you can see that the nucleus of the lymphocyte takes up nearly all of the volume of the cell. So there's very little cytoplasm visible there. So here, this dark purple is the nucleus, and this small bit of lighter color is the cytoplasm. They're primary cells of the immune system. They respond to pathogens or infected and diseased cells. They form memory cells, which we'll discuss when we talk about the immune system. And they can destroy pathogens and infected or diseased cells. Because they form memory cells, their lifespan can be days up to several years. And it takes quite an amount of time to discuss the lymphocytes. We're going to cover these in chapters 7 and 8. Next are the monocytes. Monocytes mature into macrophages and dendritic cells. They're a smaller percentage, 3 to 7%, but they're common enough that you will see them commonly in a blood smear. It's about 300 to 500 per millimeter cubed. They're the largest of the white blood cells, so you can tell them apart partly based on their size compared to the other white blood cells. They have few granules, but large digestive vacuoles. You can also tell them apart based on the shape of their nucleus. So here you can see this characteristic horseshoe or kidney-shaped nucleus of the monocytes. And you'll see that there is quite a bit of visible cytoplasm because of their large size. Monocytes are important to remove old and damaged cells. They can remove large molecular complexes, various substances, and pathogens. They help to initiate wound healing and tissue remodeling. And in the macrophage form, they will be commonly seen during chronic inflammation. Eosinophils. Eos, by the way, means pink or reddish. Oops, there's a typo on this slide. Sorry guys, let me cross that out. So let's make sure that we don't do that. That's what I get for copying and pasting. So that was for monocytes. So eosinophils don't mature into macrophages. Cross that part off. So eosinophils get the eos portion of their name because they have a pinkish reddish tone to their cytoplasm. They are granulocytes. They have large, coarse granules, and those granules are reddish in color. They have a very interesting bilobed nucleus. So 1, 2 lobes to the nucleus here. I have to tell you that this reddish in color is not quite as visible as this image shows, but that's what they tell us. Typically in a blood smear, these are so rare that they're very hard to find. They're only about 1 to 4% of the white blood cells. But you probably tell them apart based on the fact that they have coarse granules and look for that bilobed nucleus. That reddish color may be better in very nicely stained blood smears, but in sort of the typical blood smear, not always that red. Their function is destruction of parasites and viruses through their release of granules. They enhance the inflammatory response, and within their granules, they have certain chemical mediators and enzymes, for example, histamine, nucleases, lipases, and plasminogen. You will see the number of eosinophils increase during parasitic, viral infections, and allergic reactions. That includes asthma and other sensitivities. So normally they're only about 1 to 4%, and when you see them increase, you want to think about these potential markers. It could be parasites, could be virus, could be an allergic reaction. Basophils. Oh, I did the typo again. Gosh, sorry guys. Man. And then I called them eosinophils. Terrible, terrible, terrible. Sorry, I'll fix this later for you guys. Okay, basophils. Basophils are a very small percentage, even smaller percentage than the eosinophils. So it's very, very rare to see a basophil. They're less than 1% of the total white blood cell count. They're rarely seen, but when you do see them, you'll see that they're very granular. They have large granules. Those granules are different in appearance to the eosinophils. They're more purple in color. And they do have a bilobed nucleus, but because of the large size of their granules, that nucleus is often obscured. Similar to eosinophils, they're commonly seen in parasitic infections and allergic reactions. Their granules also contain histamine. And in addition, you'll see heparin along with other inflammatory mediators like prostaglandins and leukotrienes. They can help B cells to produce plasma cells, and they have high levels found in parasitic infections, allergic reactions, and asthma. So again, when you see the number of basophils increase, you want to think about the potential for parasitic infections, allergic reactions, and asthma. Lastly are mast cells. Mast cells used to be thought to be similar to basophils, but we now know based on genetic analysis that they have a different precursor origin. They do look quite similar to basophils. They're found in vascularized connective tissues near the epithelium, so you'll see them commonly in the dermis, the GI tract, and the respiratory tract. They have a very important role in inflammation. They help to increase the permeability of blood vessels, they affect smooth muscle contraction, and they regulate other cells during inflammation and healing by releasing synthesis of inflammatory mediators, in particular histamine, tryptase, cytokines, prostaglandins, leukotrienes, and growth factors. Just to show you the complexity of the mediators and the pathways for mast cells, I'm copying this figure here from chapter 7. So these are the effects of degranulation and synthesis in the mast cells. We'll cover this when we talk about the immune system and the pathways of inflammation. So if we put all of this histology together, we can look at the most common, which are neutrophils and lymphocytes, and then the less common, which are monocytes, eosinophils, and basophils. You guys want a quick way to remember these? So this is neutrophils are the most common, then lymphocytes, then monocytes, then eosinophils, and then basophils. So if we start common, will be the neutrophils, rare will be the basophils. So we have a moniker here, and it's really catchy. It's never let monkeys eat bananas. So never let monkeys eat bananas. So N for the neutrophils, L for lymphocytes, etc. And that's how you remember the order of the white blood cell percentages in your white blood cell differential. So they should be in that order, and any difference in that order can signal different conditions. For example, we talked about parasitic infections and allergy. For the cellular components, or the formed elements of blood, we also have platelets. Platelets are called thrombocytes, and they're less than 1% of the total blood volume. About 150,000 to 400,000 for the counts for platelets. They are not complete cells, they're just fragments. And if you didn't know what they were, you might just think that they were dust on your blood smear. So let's look here. Here we've got this image that has several arrows pointing to the platelets. So there's one, see that little speck there? There's another one. All these little specks are not artifact in the blood smear, they're actually platelets. They have granules, but they have no nucleus. They have a few organelles, and they actually are contractile. They have actin and myosin. They're formed from cells called megakaryocytes, and we'll talk about blood cell formation when we do our hematology lecture. They're essential for coagulation and control of bleeding, and they can release pro-inflammatory molecules, coagulation factors, growth, and angiogenesis factors. I want you to pause here and see if you can test yourselves to identify red blood cells, platelets, and the different forms of white blood cells. So take a moment here and quiz yourself, see if you can do it. Okay, did you do it? Check your answers. We had a neutrophil, an eosinophil, and a basophil. Those are all our granulocytes. Then we had a lymphocyte and a monocyte. And then sprinkled throughout, we had red blood cells and platelets. Remember to use the size of the cell, the granular or agranular appearance, and then the shape of the nucleus. Finally, there are diagnostic tests to look at various blood components. Many of these are together in the complete blood count. So we can look at the amount of hemoglobin, and that gives us an idea of the oxygen carrying capacity of the blood. We can look at the hematocrit, or the percentage of red blood cells in the blood, the total red blood cell count, the mean corpuscular volume, which is the volume of one red blood cell, and that may change in certain anemias. The mean corpuscular hemoglobin, that's the amount of hemoglobin per red blood cell. The mean corpuscular hemoglobin in a given volume. And again, these are all related to hematology. We'll talk about that in a lecture or two. And look at the variation of red blood cell size and width, that's the distribution. We can look at the number of platelets to determine clotting risk. There's also a type of cell called a reticulocyte. This is a precursor red blood cell. It will be increased with red blood cell destruction as new red blood cells will be released in their immature form. Then you can get your white blood cell counts in your leukocyte differential. And that's where you want to remember that never let monkeys eat bananas, so you can look at the percentage of each white blood cell type and when that is altered. Okay, I hope this was helpful. Let me know if you guys have any questions.